or apoptotic, or whether it is a macrovesicles. Okay. So here we are learning that this is a cell and you have a plasma membrane and we have MVP and we have these exosomes which has been secreted. So this process we learn how these exosomes are secreted. So we have different structural and biological features of the exosomes. As I said, these exosomes will have the phospholipid bilayer, and this lipid bilayer has its own biomarkers, right? Whether it is exosome or microvesicles or apoptotic, previous 2 decades before we were understanding they were waste products. But since researchers have proved that this could be used in a different therapeutic mechanisms, they've identified one. In the image, I've given CD9, CD63, 81, which acts as a cluster of differentiation to determine whether it is exosome or microvesicles or apoptotic body. Of course, we can differentiate between the cells based upon the size, but the techniques or the technology which has been present does not allow us to differentiate in a clarity to define whether it is an exosome or apoptotic or macrovesicles. So we use markers to differentiate the isolated extracellular vesicles to differentiate into exosomes or apoptotic or the macrovesicles. So these exosomes basically have been secreted by the endosomal pathway. Macrovesicles, which is basically a budding process. Apoptotic is a cell fragmentation because of the apoptosis. Okay. And the size range is 30 to 150, 100 to 1000, and 500 to 5000 for the apoptotic body. And the cargo material, basically, we need to understand what are the basic cargo material could be transported by these 3 vesicles. One, exosome, they transport miRNA, which we call as a microRNA, mRNA, then we also transport sLRNA, cholesterol, and few cases lipid molecules. Microvesicles, which we call as an ectosomes, they help in transporting DNA fragments, mRNA, proteins, and apoptotic, they help in transporting the nucleofragments and the organelles, in few cases DNA fragments also. So we know what is the major function of all these RNAs. mRNA help in messaging, transcription, translating, and then protein synthesis. MicroRNA, generally it has been applied for gene regulations. Then long coding RNA has been helpful in gene expression regulations or cellular regulations. The same way we have your cholesterol and lipid, which gives us a stability for any kind of cells, and also for travel from one part to another part, it helps. Okay, so these material will be transported from one cell to another cell as per the requirement by these 3 secreted materials from the cells, either from the exosomes or macrovesicles or apoptotic. Since we are focusing on exosome, we'll learn how these exosomes are produced in our cell. Next slide. So this is we call as an exosome biogenesis. So in this we need to understand 3 processes. One we call as exocytosis, endocytosis, and pinocytosis. Since you're all PG people, I think you know what are the 3 definitions part of it. Endocytosis is basically pulling some material from environment into the cell, we call as endocytosis. Exocytosis, whatever synthesis inside the cell will be removed and it has to be expelled from the system, and that is how it is going to remove from the system, we call as exocytosis, where you will have the plasma membrane deregulations and cytoplasm will start adjusting itself to remove as a particular vesicle. Here again it is the same process where the material from the outside cell, from the next cell, will be allowing inside the cell, so the plasma membrane again there also gets adjusted, cytoskeleton gets deregulation, and thereafter it is putting a particular secretory material inside the cell. So this is what is happening. The first will be the initiation step, the second will be maturation, third will be what is going to happen once that particular secretion has occurred, and the final one will be release. So whenever you have an initiation, we call them as budding process, where the inward budding of the plasma membrane of a particular cell will occur, and this inward budding of the will help us to form the early endosomes. Okay, so these early endosomes, whenever this process is occurring, we call them as endocytosis. Okay, and this endocytosis will allow all the molecular material, the cargo material, to diffuse into that particular early endosomes which has been formed, and that we call them as maturation, where it will be forming multivascular body. And this multivascular body will have the membrane by itself and also will have the cargo in them, which has the outer and the inner membrane, and this will also produce lot of ILVs. So this ILV is basically called as intraluminal vesicles. So whenever it is in the cell, during the biogenesis process, we call them as ILV, or we use one term called as MBB. MBB we use for multivascular body, and ILV, intraluminal vesicles. Once this is formed, it is completely defined one with a specific phospholipid bilayer, and the cargo material are fused into them. And after this, the fate is basically two processes. One, either it has to go for physiological process, or it has to go for killing itself. Either it has to go for apoptosis, or it has to release into the system to communicate what is happening in that particular cell. So either it will go for lysosome fusion, and where lytic enzymes will lyse a particular cell, enter into the apoptosis by means of apoptotic signal. And then in the case of releases, which is being released by the system, we call them as exocytosis. Okay, it is coming out of the system. So this will have, once we call them as ILV or MBB inside, when it is outside the cell, we use one term called as exosomes. Okay, and these material will help us to transport what is happening in that particular cell. So when I am diseased, that cell will have different material secretions, and my exosome is a marker to know what is happening in that particular cell. Okay, so this is what is happening in the case of biosynthesis, which is a very normal process which is occurring in a normal physiological conditions. Even in the pathological conditions or pathophysiological conditions, it is the same process where your exosome secretion will be there, and you will also have the apoptotic cells which has been produced. And along with that, we have pinocytosis. Pinocytosis basically is called as a pinching off mechanism, where it will accumulate all the outside extracellular fluid into them, and that is also pitched off. Those materials also will be misunderstood whenever we are doing the process of isolating, misunderstood as an exosome. So we need to differentiate by using different techniques and the markers to differentiate between exosome, pinocytic material, and the apoptotic materials. Okay, so this is a complete picture what is there inside that particular specific smaller exosome material. So as we know, these are the phospholipid bilayer, which we call as a double track layer, which will have the polar head group and the tail group, and will have the cholesterol material for stabilizing, and therefore it is very stable. Wherever you are giving administrative, it can easily go undisturbed, right? You can easily move from one place to another place without undergoing biodegradation. So this is one of the major points, and it will have its own mRNA, enzymes or signaling proteins, or in cases long coding RNAs or circular DNAs, RNAs, whatever it is. But the markers will differ based upon which type of exosome it is, right? Whenever we have exosome secretion from brain cells or nerve cells or lung cells, they will have different markers. But researchers have found out only 3 markers for exosomes, as I've listed already. We have CD9, 63 and 41. These are used for exosome identification. Apart from that, we have tetraspanin. So tetraspanin is again another protein which could be used to identify whether it is an exosome or a microvesicle or apoptotic material. So I differentiated for you to understand better. In the protein part, we have the CD9, CD63 and CD81 as a markers. HSP stands for heat shock protein, and we have different classified HSP there. And of course, this will help in giving a structural signaling changes which is occurring in the particular cell. And lipids, we have cholesterol, sphingolipids and ceramides. They are helpful in communication, of course, support and stability. And nucleic acids, we have miRNA, mRNA, long coding RNAs and DNA fragments. As I already told, they have been involved in genetical regulations and genetical cellular regulations. So whenever we are talking about exosome therapy or exosome biology, we need to understand that we need one donor cell and one recipient cell. So what is happening in a particular cell, I am going to understand or communicate to another cells. So this is what is going to happen in our cell in a normal physiological conditions. Something is secreted, we have MPV, this is a donor cell, and exosomes are expelled, and these exosomes carry the messages, whatever is happening inside that particular cell, and the recipient cell will start receiving the same exosomes. Okay. If it is apoptotic cell, the recipient cell will reject. Okay, it will go for inhibition. If it is an microvesicle cells, again my recipient cell will not receive it. But exosome material, my recipient cell, they have their own signal receptor mechanism, which we call as a ligand receptor mechanisms, and they accept these exosomes to pump into the recipient cells. So the exosome, which has its own messages, are pumped in, again the same process: endocytosis, exocytosis, and pinocytosis. So these are the three simple processes involved either in forming the exosome or pumping the same exosomes into the recipient cells for the messages to be carried across the cells of a particular tissue or an organ. So finally, we have a recipient cell, and this recipient cell, once it receives this exosome, they react or act according to the pathological conditions or physiological conditions based upon the cargo which has been pumped into them. So if I have X cargo into the cell of the recipient, my recipient cell will start modifying its whole metabolism based upon the cargo which has been dropped into that. So this is a basic principle which we are using to have an advanced therapeutic tool mixing exosome biology and the mRNA technology to deliver a correct genetical material or correct drug to the recipient cells. So why people are focusing on exosome biology? Because most of the labs, they have started working on exosome. This is because of only this reason, because most of the synthetic carriers which we are doing, liposome binding, then putting drug into the liposomes, then giving into the patient cell, and seeing the prognosis. Something works for few people, something doesn't work for few people. Even in the chemotherapy, few respond better, few doesn't even respond for the chemo which we are administrating. So what is the major advantage of this exosome based biology is that it can penetrate to any membrane. So it can easily penetrate exosomes into the cell. So this is basically a neuron cell I'm taking for you to explain. We have astrocyte. Astrocyte is one part of the neuron which will have its own end feet. And our exosomes can easily penetrate even through the brain. So we call them as a blood-brain barrier which can cross. Therefore, even if the person has got brain cancer or some kind of neurological disorders, it is easy for me to deliver the drug into the brain cells to have an efficient treatment. Okay, so this is one of the major things. And the next thing is, since it is nano size and natural material, nothing is going to harm our normal cells. So it is going to only target the specific cell which is in need of it. By addressing those cells, my exosomes will work. Remaining cells remains undisturbed. So this is one of the advantage, because in chemotherapy, what is happening is that if I'm giving a drug, that will affect even the neighboring cells. So people doesn't die because of cancer, they die because of the treatment or medication which has been given to them. So that drawback has been addressed in this exosome mRNA therapeutic, where it is targeting a specific cell without undisturbed, which is a natural phenomenon. So what is going to happen? People are already been working on RNA therapies, right? People have been isolating RNA, different types of RNA, mRNA, whatever the RNA it is, and giving to the recipient cells to alter the physiological process, right? So what is happening whenever I am giving a naked RNA without any biomembrane or without any stability, that will go for higher immunogenicity, because we have our own immunoreactions. My system will start accepting as a non-self material, and my own humoral and cell-mediated immunity will start competing, and therefore there will be an inhibition of the RNA which we are administrating. So whenever I am using RNA as a simple therapy, it's not 100% effective against certain disease conditions. So people have switched over to exosomes where you can deliver the mRNA correctly in a particular recipient cell. So this is what is happening here. In this case, you have lot of degradative enzymes like ribonucleases which can eat up the RNA which we are administrating, and also we call as a drinker policy where I'm giving an RNA, my cell is ready to eat it up. So we call as engulfing, eating or drinking, whatever it is, either apoptosis or chemotaxis or cell lysis will occur. Whereas here in this case of exosomal solution, that is not going to happen because my RNA I am going to completely pack in the lipid bilayer. It's basically a completely packed, which is a box that lipid bilayer cannot be disturbed by my enzymes. So this is one of the major advantages of using exosomes, and degradation is not at all caused by the RNAs, and the stability is more. And once this mRNA is designed by our lab, or when we are designing according to our convenience, then it enhances the therapeutic efficacy again. Right? It is not needed to have an mRNA of the cell as such. I can modify, genetically engineer, reconstruct the plasmid and insert it into the exosome, and what I need, I can address in the particular cases. So these are the major advantages: superior safety and biocompatibility. Exosomes, the clearance is very easy. So when I am taking exosome as a drug or as a medicine or as an IV injection, clearing off from my system is very easy because it undergoes normal physiological clearance, debris clearance. But when I am taking a drug, synthetic carrier, removing a drug from a particular cell takes months to gather. Very good example, paracetamol. If I am taking paracetamol today, it takes at least 1 week or 2 weeks to be removed from my cell. Antibiotic, it takes at least 6 months to be completely removed from my system. But exosomes, in that case, it is not like that. It's very easy to clear off from our system faster. So it takes only 2 to 3 days to be removed from our system as an apoptotic cells. Toxicity, it shows only mild effect. Whenever it has been administered in an animal model, it shows only a mild effect. But of course, it is ignorable because it is a synthesized material. It's not a synthesized material, it is a natural derived material. And immunogenicity is very low because system understands as a self material, not as a non-self material. Biocompatibility is also more. All these things are adverse or inverse in the case of your synthetic carriers. So this is you class from biotechnology or microbiology or biochemistry, you will be wondering why I need to know about exosomes. You need to know only few couple of techniques to isolate an exosome. But it's a very difficult task. My research people are doing on exosome isolation. It takes nearly 6 months to only isolate, and thereafter to differentiate what are they. That takes another 3 to 4 months. But once you isolate the exosomes, that is the way we can have the applications part more interesting, right? At least it takes 1 year to validate. But the process is as simple as it is. The first one is designing of the DNA template. Okay, it is not necessary that I should have the common DNA. According to my convenience, I can redesign, re-engineer my own DNA template. So I'll be having my own gene sequences wherever possible. I can manipulate or I can correct it. I'll have my own DNA sequences to have an efficient protein synthesis. Finally, it is going to have an mRNA, then translate into a protein synthesis. So whatever protein I want to synthesize, accordingly I can reconstruct my mRNAs. Okay, so that is the first step. Second step will be in vitro transcription. In vitro transcription basically where we use mRNA synthesizing in the laboratory by using either vector technology or by means of plasmid constructions. Once it is done, we do the structural modifications of the mRNA, of rearranging the protective caps or the UTR, which we call as an untranslated region, or the CDS, the coding sequences, and long chains of adding or removing the nucleotides. So we reconstruct in the laboratory to have our own mRNA, after which we check whether it is of purity, and then finally we go for larger scale. So we produce in a smaller scale, check the viability, what is happening. whether it is able to produce a protein or not. Once it is done in a small scale in a test tube level, we translate to the larger scale to have a higher bulk productions. So for this, what are the major things which we need to know is the techniques, plasmid construction, how does an MRNA will work. The technique, what you need to do is only Western blotting. We have an NTA, we call as a track analyzer to analyze whether these exosomes are having the markers or not. Then marking technology to understand whether it is in microvesicle or apoptotic, where you need to have knowledge on spectrophotometer or calorimeter, or simple techniques where you can read the nanometers, filters, using filters. So these are the basic techniques, and along with that PCR to know how it is going to give the protein synthesis. So in our lab we are doing two processes. One we call them as endogenous loading to produce the exosomes, and we also produce from the exogenous loading. Two processes we are using. And you may understand that it could be produced only by the cells. No, we can take any plant, you can take any flower. I like a jasmine flower, I can take that. From that also I can isolate exosomes. I like different sources of microorganisms. I have a culture of microorganisms. From the organisms also I can I can isolate the exosome. The source can be completely different. So people have identified different origin of exosome isolation. People are now focusing on exosomes when compared to the mammalian exosomes, because mammalian exosomes we have lot of ethical issues. You can take any kind of phytocomponent, start isolating the exosomes. We do in two loading. One we call as endogenous, and another one we call as exogenous. Endogenous, what we do is we usually select a donor cell, program that donor cell, and according to the program what we are expecting, we redesign everything. We re-engineer everything. Either let it be a cargo material or let it be an mRNA or whatever it is. So everything is designed by us. So nothing is happening from the sources. We don't get it from the sources. I decide this lipid should be into that box. This nucleic acid should be into that box. This cholesterol moiety should be into that particular box. I decide my own phospholipid bilayer. Then I design my own mRNA for initiating. So this is what is happening. Very good example, or what we do is, first we transfer to the engineering plasmid, select the donor cell, where we design everything by ourselves, and allow it for incubation, where it produces its own ribosomes because of the cargo which we have put into that particular exosome, and has its own mRNA. Finally, it does its role, and you get the synthesized protein to be delivered in that particular donor cells. So again, for this, the real challenge lies in fusion of the cells. We can produce exosome, we can produce everything separate. But putting that cargo into the exosome is very difficult. Most of the cases we fail whenever we are characterizing using the techniques to understand whether the cargo has been loaded into the exosome. It's very difficult for us to understand. But out of 100 cases, at least 2 succeeds. For 100 doing, we need to at least spend 7 months to 8 months. So this is one of the endosome loading, which we call as a preloading. Exogenous loading, anyone in any laboratory where you have a simple ultracentrifugation technique, or when you have a pipette, that is when you have a ultracentrifugation vials, it is very easy to do. And when you have a high speed end with freezing temperatures, it's very easy to isolate. But the problem is to differentiate whether it is an exosome microvesicles. That you need to have a cost-end equipments and high-end equipments and the chemicals. So what we do is we have 4 processes. One is incubation. Incubation, what we do, we take any sources and start centrifugation and collect the exosomes. Once the exosomes are collected, we put it into the along with the drug of choice. For example, I wanted to put in the paracetamol. So I'll take a paracetamol, infuse the exosome with the paracetamol, and my paracetamol should be into the as a cargo into the exosomes. So for that, we will have the different techniques to understand whether that paracetamol has been infused in the exosomes. Instead of giving paracetamol, I give exosomes, right? And these exosomes are given to the patient or the donor cell, where they go and attach to the targeted site or mechanism of action particular cell, not to the overall cells. Okay, so this we call as an incubation, and this is done within 24 hours to 72 hours, 2 days. Within 2 days to 3 days, the process is complete. We have exosome liposomal hybrids where we fuse the liposomes or exosomes with the already existing lipid material, and we have a double protective coat. Along with that, we'll infuse with the cargo. So this we have the exosome liposome hybrids because most of the cases whenever we have non-exosomal material, when adding lysosome, these lytic material can lyse and enter into the apoptotic. So when I'm getting a purified material, I'll be getting only the exosome. So what is the point of having the lysosome is that to have a specific exosome which is functioning efficiently, not the other material. For that we are using this lysosome hybrid material. And electroporation will take everything in the vial or in a beaker and just give an electrical field, and they either temporarily or continuously. Usually we give a break because the membrane should not break. So we have half an hour break, then half an hour break, half an hour break. So we call them as electroporation field, and we start synthesizing or producing the drug infused exosomes. And last will be sonication. Again, this is basically high frequency sound, and this high frequency sound will be very low decibel, where the fusion of the drug with the exosomes can take place. In all the four conditions, our thought process is that the biomembrane should open a little and my drug should pass through. So in few cases we'll have a passive diffusers. In few cases we add certain chemicals to facilitate the process so that my drug enters as a cargo into the exosomes. So these two methods we can produce in vitro for producing the exosomes. So this is what happening, workflow from the lab to the war field. Okay. So in our lab we are selecting particular cells. Finally we are producing it. Purification, we use separation, isolation and purification technique, and finally we go for fractionization. And in vivo administration, either in the rat model or rabbit models or any other zebrafish models, and test the exosomes efficacies. Finally, when targeted into the target cell, how the delivery is, because we are talking strongly only about the delivery, not about the administration. If any drug could be administered, how it is safely delivered to a particular site of cell, that will be monitored here. And finally, the therapeutic effect of the exosomes will be understood. In the therapeutic, there are three parameters. We need to understand the biodistribution, whether it has been distributed from head to toe, or what is the outcome of it. If I am giving exosomes, whether they are developing long-term or short-term lethal effects, so that will be also monitored. And pharmacokinetic studies and pharmacological effects will be also understood in the sixth step. Finally, whether my immune system is getting altered or activated, or it is going for suppression. So that is the major part whenever we are working on the exosome mRNA therapeutic, because we till 6th it is fine. When it is going for 7, it gives us an adverse reflex whenever the mRNA is not matching with the target axis. So this is a common treatment for cancer, right? Since we are talking about how it is going from discoveries to the clinical side, in the discovery part it is very minimal process, but when it is in the application part is very part. In the cancer, we have 4 therapies which we call. One, diagnosis. Usually people diagnose using blood sample or the plasma or the serum sample, where the exosomes can be derived exosomes from the particular cancer cell, and the markers will be understood. Based upon the markers, we can redesign the whole structure. So this is one way people are working in the cancer, by understanding the liquid biopsies. Then next comes your immunotherapy. In the immunotherapies, they give different immunocompetent materials or immunosuppressive material, and then later on go for whatever the drug which has to be administered. So we call them as immunotherapies. And few people are focusing on gene silencing, where siRNA, single-stranded RNAs can also interfere with the RNA, and they also can reduce the spreading of the cancer. Okay. And the last one will be the tumor engineering. There is engineering of the mRNAs where it could be used as a therapeutic one. So in a cancer, we are having 5 to 6 different types of therapies, which is still working in the day-to-day life. But the last 3 to 4 years, completely switched over to the precise medicine. What is precise medicine? I will differ, I behave differently when compared to the other people. For everybody, I can't give the same medications, same molecule. So it should be highly specific. For that, people are focusing on the exosome mRNA therapeutics. So this is what is happening in the cancer. The first one we call as a targeted therapy. In the targeted therapy, we create or we engineer the extracellular vesicles with a defined mRNA and put it into the recipient cell. Here the recipient cell for us is cancer. So in the cancer cell, we design the mRNA and this mRNA is dropped into the recipient cells and synthesize certain set of protein, and those set of protein will stop the or inhibit the growth of the cancer cells. So this is very highly specific. Toxic level is very low and the adverse effect is also very low. So people don't prefer nowadays to go for chemotherapy or chemo drugs, and we have an advanced therapy called as photon therapy where they shot on the particular cell. So they are trying to fuse this exosome with the photon shots also, where it could be labeled with a radioactive labeling and it can shot on that specific site where it is manipulating more or it is growing more. So it will enhance all the cellular regulatory proteins like caspases and other things, and this will be inhibited. The second will be your engineered EVs, which could also focus on the immunocells, right? You know that dendritic cells plays a very important role in clearing of the unwanted cells. So they engineered mRNA and put it in the EV. They target the working cells. What are the working cells? We have macrophages, NK cells, dendritic cells. They have their own pseudopodia and consider the recipient cell as a non-self material, start eating, engulfing, and then a normal apoptotic process will carry on. Third will be liquid biopsies, which is now recently people are also have been using, and they'll have their own specific markers which has been circulated based on the specific markers. We design the mRNA loaded exosomes to target those things. Okay, very good example. This pancreatic cancer is very difficult to be diagnosed, and people now use this exosome markers to diagnose the pancreatic cancer. Very good example is your glypican, which we call as GPC1, which will be elevated in our blood, which will be circulated across the cell. So when we are using this exosome anti-markers to define where is GPC1, it is easy for us to trap them in the initial stage. I think few people will have more interest in doing research or taking yourself to the next level in the exosome biology. There are a lot of research papers where they have been publishing frequently, and this is one of the paper which talks about tumor-derived exosomes. So what they are doing is, they are taking a tumor cell and from there they are isolating the exosomes, and those exosomes morphological and characteristics they will study. And according to the characteristics, they design our own exosomes and put it in the normal cell to have an anti-effect. So this paper completely gives us a complete picture about tumor and cancer derived exosomes, how to synthesize, how to produce and everything. And again for immunotherapy and also to produce different vaccines against cancer based on exosome therapeutics. This is in the glioblastoma. So glioblastoma is basically, we call it as brain cancer. Okay. So during the brain cancer, we have a lot of side effects. Basically, they will forget people. They have a lot of oxidative destruction. Neuroinflammation will be more. So we have a lot of symptoms. In normal conditions, what is happening? We'll have the cold tumor environment. And these cold tumor environment, whenever the person is having the brain cancer, the immunogenicity will be very high. Means they will be resistant to most of the they will not be resistant, they have a suppressive materials, and the antigenic presentations also will be very low. Means the MHC molecule which picks up the antigenic fragment which is giving to the T cell will be very minimal. So my immunological response or the tolerance will not be as high as in the normal cases in the case of your brain cancer, which we call as a glioblastoma. So what we are going to do is, we are going to design one IMSEV. What is this IMSEV? We call it immunogenic small extracellular vesicle. And these extracellular vesicles will carry the mRNA of my engineered material and will be administered to the tumor cells in the brain. We already saw that extracellular vesicle can pass through even brain without undetected, like a GPS tracker. So we design our EV where mRNA, designed mRNA, and give it to the targeted cells. So what happened was the cold environment translated into the hot environment. What is hot environment? Where all the cells which is involved in the immunotherapies will be playing an important role. T cells will be picking up the antigen represented by MHC1 material and either MHC1 or MHC2. B cells started maturing more and they started again acting its own tumor cells. Means I am doing my own process to initiate or to enhance the environment and depress the tumor mechanisms. Basically, we are putting an exosome to inhibit the production of the tumor or spreading of the tumors in that particular area. So this we call as inflamed one, hot one. This we call as the cold one. In the hot one, we can see the T cells. M stands for macrophage cells. MHC, which will represent the antigen to that, and there my own cells are destructing the tumor which has been present in them. Okay, so this is one of the turning point in the therapies. So this paper is giving about liquid biopsy tool to understand the brain cancer and also microenvironmental conditions where you can change just by changing the pH temperature and the buffering capacity of that particular cell can also alter the exosomic delivery. So this paper completely gives us about the picture of the glioblastoma, how it could be diagnosed, how the exosomes-based material can be used in the treatment of brain cancer. So this is about biomimetic therapeutics. If at all you wanted to land up in any research domain, I suggest you to go for a biomimic lab. Biomimic lab is basically we don't synthesize anything in the laboratory synthetically. We take everything from our own cells, which is basically mimicking the actions. So our EV has its mimicking capacity like that of the virus. Usually when you see a virus, it has its own, internal cargo, which we call as the nucleic acids, and we have an outer shell, and we also have the ligands. The same way, I can also design my own exosomes where it can have its own RNA material, own nucleic acids, and the phospholipid bilayer, and the extracellular vesicle. So I can design an exosome just